Keynote. It is a delight to introduce to you Dr. Barney Graham, Deputy Director of the Vaccine Research Center, NIAID, and Chief of the Viral Pathogenesis Laboratory, NIH. Obviously, there is no bigger story this year than the race to develop the COVID-19 vaccines. We are very excited to have Dr. Graham here to give us the NIH perspective on this. We know much of it happened there and was supported by NIH, both extramurally and intramurally, but also there was a huge amount of leadership and work that was done there. And I'm looking forward to hearing about it. Dr. Graham's laboratory has developed novel vaccines for RSV, influenza, Zika, and coronavirus, including the first COVID-19 vaccine and monoclonal antibodies to enter clinical testing and that have now achieved emergency use authorization. Dr. Graham's research interests are vaccine development for viral disease, viral pathogenesis, mechanisms of immunity, pandemic preparedness. Dr. Graham has a BA from Rice University, an MD from the University of Kansas School of Medicine, and a PhD in microbiology and immunology, Vanderbilt University School of Medicine, where he completed his internal medicine residency, chief residencies, and his fellowship in infectious disease. We could not have asked for a better prepared scientist at the helm at this time during this terrible pandemic, nor a better person to talk to us about it. Please welcome Dr. Barney Graham.
Thank you, and it's my pleasure to be here. This is a group I feel like I have a lot in common with, so bear with me while I get this going again. I'm going to tell you a little bit about the backstory. I think you heard some of the trial results yesterday, and so I'm going to spend most of my time talking about how we got started and maybe where this can take us into the future. I work at the Vaccine Research Center, which was founded in around the year 2000 to work on an HIV vaccine, which we obviously don't have yet. But the work that's been done to try to make an HIV vaccine has generated technologies that have addressed a number of unmet medical needs, and including emerging infectious diseases as listed here. And that's largely because we are privileged as a division on the intramural campus to have not only this basic research building, but about 100 engineers in Gaithersburg to do process development, another 100 or so to do pilot GMP manufacturing, a self-contained clinic within the clinical center, and a laboratory devoted to doing analysis on those clinical samples. And it's run by a relatively small group of PIs and program heads led by John Moscola, pictured here. Over the years, we have used a number of different approaches to deliver vaccines, including nucleic acids that I'll mention today. We've made adenovirus vectors for Ebola and HIV, virus-like particles for chikungunya. I'll mention the protein work on RSV briefly. I won't mention the nanoparticle program for supraseasonal influenza vaccine development. But I will mention monoclonal antibodies, and these products have been sent all over the world. And this last year, we had our first approved product, which is the MAB114 that was developed to treat Ebola. And in 2018 in Eastern Congo, that was shown to effectively treat and reduce mortality in Ebola. And that drug, if you ever need it, is called Ebanga. So we're faced with emerging pathogens that keep coming at us, and this is a brief list of some of the ones I've encountered during my career. And I just want to point out that most of these have started as regional problems and then either spread, and they start from a zoonotic or vector-borne source and then spread around the world as a pandemic threat. And even HIV, which has developed a life of its own, originally was a regional, zoonotic, emerging infectious disease that was not recognized in time to prevent it from being a pandemic. And so the public health needs for emerging viruses, vaccines for unmet needs, and improving licensed vaccines is not been met by traditional approaches that are listed here, like passive surveillance, contact tracing, and quarantine. But as I mentioned, there are a number of new technologies that have changed the way we think about vaccinology, especially over the last 10 or 12 years, that allow us to take a more engineering approach and have more reliable timelines and milestones. In addition to making better vaccines, we have to learn how to go faster. And in 2014, we responded to this public health emergency of international concern, the Ebola crisis in West Africa. We had a vaccine on the shelf ready to start a phase 1 trial and moved it as quickly as possible. But as we got it to phase 3 in Liberia about a year later, that epidemic had already waned. And so since we weren't in Guinea, we didn't get an answer. In 2016, on another public health emergency of international concern, we made a DNA vaccine for Zika and had a phase 1 trial started within 100 days. But again, by the time we got to Central and South America in an efficacy trial, the epidemic had waned and we did not get an answer. So we need to go faster. And the way technologies have been used in the past for vaccine development has been opportunistically. And even though these first few, the smallpox, rabies, and yellow fever in particular, were really done by the will of individuals, this next cluster of vaccines, most of the live and inactivated whole virus vaccines, were developed because Enders developed cell culture and it allowed the growth of viruses in vitro. This next cluster of vaccines came with advances in molecular biology and the ability to make recombinant proteins and virus-like particles and reassortant viruses. And then the new technologies are just coming online, and I consider the first one the VSV recombinant vector-based vaccine for Ebola that was gene-based, an example of gene-based antigen delivery. And we thought maybe RSV would be the first example of structural biology being used as a structure-guided vaccine development. But coronavirus slipped in between, and it has used structure-based design, rapid manufacturing, and number of other technologies to make this development process feasible. And so these are the new technologies, and I'm not going to review them again, but this top group allows you to have more precise antigen design, and this bottom group allows you to go faster. And I think the combination of precision and speed is what we need for pandemic preparedness and response. And I think of it as the preparedness being the long-term process of knowing how to design the right antigen or deliver the right thing to make the right immune response, and the response being these new platform approaches that can allow you to produce a product and release it more quickly. So our thinking about this started really around respiratory syncytial virus, which in 2013, after several years, we were finally able to capture this pre-fusion conformation of the F protein as it sits on a virus membrane. And during that, it revealed some epitopes at the apex of this molecule that were highly neutralization sensitive and hadn't been recognized before because this molecule is metastable and rearranges even spontaneously on the virus surface. And those epitopes are not present on this molecule, which is the one that's been used repeatedly in clinical trials at least 5 times over the last few decades with the same result of boosting neutralizing activity by about 2 to 3-fold. But if you can stabilize this conformation with the C-terminal trimerization domain and internal disulfide and cavity filling mutations, you can now, with a single dose, even without adjuvant, induce up to or more 16-fold increase in neutralizing activity. And this now becomes a vaccine that's potentially usable in either for maternal immunization or in the elderly. And just to emphasize this process that class 1 fusion proteins go through, these are the molecules responsible for virus entry into cells, and they are what is required for membrane fusion and entry of this nucleocapsid and genome of the virus. And if you immunize against the beginning functional part, you've got a vaccine. And if you immunize against this non-functional part, 6-helix bundle at the end, you have a vaccine that will make binding antibody, but not very good functional antibody. So thinking across virus families and that have these class 1 fusion proteins that are many of the envelope viruses of interest, the RSV and pneumovirus is very similar to the F protein in paramyxoviruses. And this other cluster of influenza, HIV, Ebola, and Lassa have class 1 fusion proteins, but they're slightly different with a different domain capping the fusion machinery. So this domain has to be removed in order for the fusion machinery to be revealed. And then coronavirus has all of this. It has this intervening segment and the cap, but all of them have the similar property of endoproteolytic cleavage. This fusion protein is released, the heptad repeats fold back and pull the membranes together. And so they all despite the different shapes and topography, have a very similar function. And you can use a similar strategy with these different proteins if they're the target for the vaccine. And so after our experience in 2016 with Zika, not getting there fast enough and thinking that we could start developing generalizable approaches across viruses and selected viral families, we made a deal with Moderna in 2017 to do a pandemic preparedness exercise on the paramyxoviruses and coronaviruses using Nipah as the prototype for paramyxoviruses and MERS coronavirus as the prototype for the coronaviruses. Paramyxoviruses, as I said, have a similar fusion protein structure to RSV, but they also have these attachment proteins that are either HN, H, or G, depending on their hemagglutination activity. And in different viruses, one or the other is the better vaccine target. In some cases, this attachment protein is a better target. In other cases, the pre-fusion form of F is a better target. So we have spent time making the antigens for the pre-F and the attachment proteins. And for Nipah, measles, and mumps, we now have stabilized pre-fusion examples of the fusion protein. And we've learned how to make these chimeric molecules in which the pre-fusion F is attached to 3 protomers of the attachment protein. And so now you can make a single molecule that contains both the pre-fusion that has the neutralization sensitive sites on it and the attachment protein in one molecule. And we've now delivered this by both protein and mRNA and shown that you can protect ferrets from lethal challenge with Nipah and induce good antibody responses to all of these viruses. For coronaviruses, this was motivated in part because the MERS coronavirus was coming at the same time we were finding this pre-fusion structure of RSV. And so as that project moved into the clinic, we and as Jason McClellan, who worked with me on the RSV structure, he was a fellow in Peter Kwong's lab at the time, but was moving to his new faculty position. We decided to start a project on coronaviruses and try to do the same thing. And so it took a few years. We finally got the structure of the spike. And then the year later, we figured out how to stabilize the spike in its pre-fusion state and show that it was a better antigen or better vaccine when it was delivered that way. And then we showed that those stabilizing mutations, which involved putting two prolines between the central helix and the heptad repeat, this prevented that the extension of that heptad repeat with the fusion peptide on it and prevented it from stacking here to put it into the cell and start the fusion process. So this largely stabilized the spike protein of HKU1, but then it also stabilized the spike proteins of MERS and SARS and now a number of other coronaviruses, allowing us not only to have the apical epitopes preserved on this pre-fusion structure, but when it was stabilized, it also turned out to have better protein expression from transduced cells. So for instance, the MERS stabilized spike produced 50-fold more protein than the wild type version of the spike, which may be an advantage for gene-based delivery. And then it also allowed the solution for structures of these other spike proteins for MERS and SARS and for alpha coronaviruses, veterinary coronaviruses, and other endemic beta coronaviruses, and even pre-emergent bat-derived coronaviruses. So this solution for stabilizing spike provided a lot of structural information. It was the basis for making an mRNA vaccine with Moderna that was tested in mice. We showed that 0.1 or 1 microgram doses could completely prevent virus replication in the lung and also prevent weight loss from this lethal infection in the human DPP-4 transgenic mice that were developed by Ralph Barrick. And in Ralph's lab, who had shown a vaccine-enhanced illness syndrome in the SARS-1 work that he had done before, we went down to limiting doses of this vaccine and showed that even the 0.01 microgram could reduce viral load by a log, but there was still a lot of virus replicating. And in that setting, the question was whether this partial immunity would cause partial protection, which it did, or would it cause the vaccine-enhanced illness? And so in this case, the partial immunity led to partial protection, and we chose mRNA in part because of our prior work on RSV that suggested that pattern of immune response that mRNA would make would avoid those signals of Th2, IL-4, IL-13, and eosinophilia that was a marker for the vaccine-enhanced illness. So when the coronavirus showed up at the end of 2019, and we found out it was a beta coronavirus on the 6th of January, we had been planning to take the Nipah chimeric molecule as an mRNA into the clinic with Moderna as the demonstration project for this pandemic preparedness. But at that moment, we decided to switch back to coronavirus, and we're glad we did, because this coronavirus that we have circulating is now one of the deadliest respiratory virus pandemics in history. It's larger than the Asian flu in '57 or the Hong Kong flu in '68 that most of us lived through. And only the Spanish flu or the 1918 influenza pandemic exceeds the size, as we're almost up to 3 million deaths. That was at a time that we didn't even know it was caused by a virus. The influenza virus wasn't discovered until the 1930s. So here are respiratory epithelial cells that are infected with SARS coronavirus 2. And you see these viruses coming out of the ciliated cell, some of them trapped in mucus here. And looking at a larger view, you see these spherical objects, and they're about 80 nanometers in diameter, and they have these knobs on the surface, which is the spike protein. And so the spike protein is the prominent feature on the surface of the virus and is the main target for vaccine development. And as a testimony to the way technology has changed, the WHO landscape analysis has shown that there's over 250 vaccine development programs in the world, which is remarkable. And so, almost 80 now, I think, are in clinical studies, and many of them have gone through phase 3 testing and are in emergency use around the world. In this country now, nucleic acids from Pfizer and Moderna are in emergency use, and the J&J Ad26 vector is in emergency use, but Novavax is coming along soon, and so is AstraZeneca, and then whole inactivated virus vaccines from China are being used now in Brazil and a few other countries. So this went fast during the year, but it was because of this prior information. And just so by the time these sequences were released on the 10th, we designed products and reagents on the 11th of January, and Moderna, because of our prior relationship and prior agreements, decided at risk to start manufacturing immediately. They were able to give us a clinical grade product back in 41 days, and that allowed the phase 1 trial to start within 60 days. The other reagents were used to make lyses, to solve structures, and to confirm immunogenicity in mice. And that was only possible because of this prior work on coronaviruses that we had done over these last 6 years, and that was informed by the work on RSV. It was guided by some of our thinking on pandemic preparedness that we had done, thinking of prototype pathogen preparedness for across virus families. It was motivated by our inability to get to the Ebola and Zika fast enough. And then one of the byproducts of this new technology is that the same reagents that you need for vaccine development can be used for antibody discovery. And so taking the protein, knowing that it was in the right conformation, we sent a B-cell probe and convalescent PBMCs to our collaborator Epitopes that we've been working with to look for cross-reactive coronavirus antibodies. And we then screened some of the antibodies they found for neutralizing activity, and Epitopes made a deal with Lilly, and Lilly rapidly moved those products into phase 3 testing and ended up with this, what we call MAB 555, they called bamlanivimab, which is now under emergency use authorization for treating people with mild to moderate coronavirus disease and reducing the frequency of progression to hospitalization. And all of this really I attribute to the work on HIV, so far the unsuccessful work on making an HIV vaccine, but all that effort to make these new ways of thinking and making reagents for vaccines has been invaluable. So once you have the protein and you know it's in the right conformation, you can make diagnostics, you can discover new antibodies, and you can make vaccines, as we did with Moderna. And these other manufacturers who are doing studies in the US, at least, the Pfizer, AstraZeneca, and Janssen, Novavax, all but AstraZeneca are using the stabilized version of the spike using the 2 proline mutation at the top of the central helix. So that prior information has helped everyone go faster. The mRNA that was used for the Pfizer and Moderna both use the same type of mRNA, meaning that it uses a 1-methyluridine modification in the RNA in order to reduce the amount of signaling that goes through TLR7, TLR3, and these cytoplasmic detectors that lead to type 1 interferon production. And so using 1-methyl-uridine, Drew Weissman and Caitlin Carrico found about 15 years ago that this would reduce this signaling and allow protein expression to go on a little longer. So as this mRNA comes into the cell, protected by a lipid nanoparticle, it's a combination of 4 lipids that prevents degradation, but also helps deliver it into the cell. This protein is designed to be a membrane-anchored, full-length spike protein to induce these antibodies that we now know can be protective. So this is a combination of both the antigen design and a special mRNA technology that effectively produces protein. And over these last few months, we've been able to show in mice and monkeys and humans that it's very immunogenic and is inducing high antibody responses. This is live virus neutralization in monkeys compared to convalescent sera in humans. And in the human studies, we were reaching neutralizing antibody titers that were in the upper quadrant of convalescent sera. We showed protection in both lung and upper airways in both mice and monkeys. We've been able to show that the T cell response is Th1 oriented, so it's not making any of the IL-4, 5, or 13 that has been associated with the vaccine-enhanced illness in the past. And the mRNA has worked surprisingly well in older age groups. So in middle-aged adults up to 70 or older adults over 71, the antibody responses were very comparable to what was seen in the younger adults. And the T cell responses were also comparable and similar in that they were Th1 oriented. Now, this is data that isn't quite published yet. It's close, but these are the 3 age groups in columns: the younger adults, middle adults, and older adults with binding antibody across the top, pseudovirus neutralization in the middle, and live virus, whole virus neutralization at the bottom. And these are antibody assays over 6 months after the second dose. You see that the antibody is maintained fairly well, even across age groups, and especially in the live virus neutralization, which we think is the most aligned with what is going to be a correlative protection. You have relatively good maintenance of antibody activity over 6 months, at least. And that results in these efficacy curves. I'm sure that you heard about this from Dr. Gruber and also Moderna yesterday, but this is the accumulative incidence of infections in both the Pfizer placebo groups and the Moderna placebo groups compared to the vaccine groups that remain very close to baseline, resulting in efficacy against symptomatic disease of about 95%, but so far 100% efficacy against severe disease, meaning low oxygen saturation or hospitalization. So this was a very gratifying result. And I think Dr. Gruber may have shown you this image from our days at Rice. It's been a remarkable journey for Bill and I, who are roommates at Rice University. And he's now, as you heard yesterday, the Vice President of Clinical Research at Pfizer, and has managed most of the studies you've seen from Pfizer. And now Rice Magazine made this little image of us still working together. But it's been an interesting year for me to see and interact with Bill more than I have for many, many years. And the vaccine rollout is now, as everyone knows, we've been able to immunize our frontline workers. These are many of our staff members, colleagues, and friends. This is Dr. Kizzmekia Corbett, who is a research fellow in my lab, who's been central to getting the work done during this last few years with the Moderna collaboration and managing much of the preclinical work during this last year. And Dr. Moscola, our director, is here. And we're celebrating vaccination. This is the vice president after her second dose. President Biden visited to learn more about this process, and family members and other friends are all getting vaccinated. So I hope that's also true in your families. So in summary, the rapid pandemic response was based on prior fundamental, basic, and translational research. And I think that to me is the main message here, is that without that prior knowledge and prior work and preparation, we could not have gone as fast as we did. And with more prior work, if we'd already done, for instance, a phase 1 trial with MERS coronavirus vaccine with mRNA, I think we could have saved about 2 additional months and been able to start vaccinating in October instead of in December. So I think even more preparation could be helpful. It's important to have both precision and speed. One without the other is not going to help you very much. And these pre-established public-private partnerships were essential for both vaccine and antibody development, which, as you know, are now authorized. We think this is the proof of concept for the prototype pathogen approach for pandemic preparedness that we started with in this collaboration. And so here's the PIs again: Jason McClellan, who's been involved in most of the structural work over these last 10 years with us, and Dr. Corbett is here. Moderna, Vanderbilt, and UNC were important academic collaborators along with Dr. McClellan. Mark Dennison at Vanderbilt, Ralph Barrick at UNC were very important in those early days for both the animal and the human studies. And then all these groups were involved as well, and particularly another extramural division of NIAID, DMID, held the IND for that initial phase 1 trial and used their clinical trial networks to start that phase 1 trial. That was important for supporting the IND and advanced development of the subsequent trials. And then just to thank all the other people at the Vaccine Research Center who pivoted off of their usual work to help with this coronavirus program. And it's almost an uncountable number of people who've been involved in this. And I hope you saw longer lists of acknowledgements for all the Coronavirus Prevention Trials Network, who have now enrolled almost 200,000 people in this collection of phase 3 trials. So I will stop there and take any questions you might have.
That was fantastic. Thank you very much, Barney. I know there'll be lots of questions. And Arthur, are you raising your hand?
Yep.
Okay.
I am. So that was a beautiful lecture and I really enjoyed it. You know, the one little kind of wrinkle I would say is we still have a little bit of a movement to do in study sections and so on, because I've been very much involved or knowledgeable about Wiseman and Conico's work, and they had all kind of trouble getting funded in the late 90s in order to make the stabilization of the mRNA. And there were reports, you know, like Yellow and Burson published theirs, saying this will never work, it's too adventuresome, there's not enough preliminary data. So we just have to, in all the celebration, which is wonderful, and that was just a great lecture, I would just say that all of us need to just be aware that we need to take some chances, particularly on young, not totally established scientists when they come up with great ideas. And that's also a lesson to learn in this fantastic development. So I thought I'd just mention that and see the response.
Yeah, well, thank you. You know, for me, what that raises is the value of the intramural program at NIH, because it is what gives us the privilege of taking risks and doing things that may or may not work. And
Right.
so to me, the intramural program at NIH is a very special, privileged place to be working. And it's one, you know, and I was an R01 investigator at Vanderbilt before coming to NIH, so I know that struggle, and I still have a lot of friends in that struggle. And in fact, one of our, I think, better ideas for how to make better flu vaccines, in a collaboration with an investigator at University of Washington, he's having a terrible time even getting his grant past into the review. It's been triaged twice now. So I think we do need a little more imagination in our study sections. I could wholly agree with that.
Thank you.
I think I have a question or two, first from Mauricio and then Barry, and I'm sure there's another pile too. Go ahead, Mauricio.
Thank you. First of all, fantastic presentation and fantastic work. But I think you pointed out that, you know, that the success really was due to kind of work that preceded this pandemic, and particularly by a private-public partnership. And we find so many investigators that are afraid of working with industry, that they feel that they'll be tainted by working with industry, that somehow, you know, their science will be frowned upon if they outreach to industry. And yet, you know, frankly, we wouldn't have any of the vaccines if it wasn't for the critical collaboration with industry. So how do we change the narrative that makes so many PIs allergic to interactions with industry?
Well, it's complicated for us in the intramural program. I don't think it's commonly known, but it's essential for us to have industry partners, or all of this work gets stuck in the journal. We write a nice paper and it's in a journal article, but it doesn't go anywhere. And so if you really want, in this country, the system we have at least, if you don't have an industry partner, you are unlikely to get past phase 1. And so that's the reason we try to bring industry partners in early in all of our development programs. And you know, it's the VRC is a little different than a lot of other NIH, which is more focused on basic research, but we were established largely to be translational and try to make products, which is not necessarily the goal of every NIH lab, but it is of many of the labs. And in our system, you cannot do advanced development without industry partners. And I think that model can work, but I also think we need another public option for things that are not necessarily commercially viable, because industry can't do everything. They have to stay in business. And so we need more public options, or public philanthropy or public nonprofit type of partnerships to do some things that cannot be done otherwise.
Great. Barry, I think you were next.
Yeah, so I'll ask my question and I'll make a comment about what you just said. My question is a little bit off to the side, but I want to first start by expressing my deep appreciation and admiration for what you've done. I mean, it is really one of the most spectacular stories in the history of medical science. And I'm not to hyperbole, but this is really an astounding accomplishment. So my question, however, is a very mundane one, which I keep getting asked, which is, when do you think there'll actually be a full approval? And the reason I keep getting asked this question, although it may not be known by everyone, is that an institution can't require vaccination as a condition of employment until there is a full approval. And there's a lot of discussion about this issue. So I was just wondering if that is, you know, obviously if it's confidential information, I don't want it disclosed, but I am getting asked this question a lot.
Yeah, well, I think the general parameters are not necessarily proprietary, but you know, part of the problem with going fast, even though I think this was one of the best data packages I've ever seen, even though it was put together in about 10 months. But some things aren't done, like immunization of special populations, like the children and pregnant women and breastfeeding women, immunocompromised people. So some of that is still being done. And so a lot of the licensure BLA application is waiting for more safety data in more specialized populations. And I suspect that those applications will be in mid-year and maybe approved by end of year, but it's a lot of that is more dependent on gathering a larger safety database to make it easier for the FDA to agree.
Great. The one comment I'll make, years ago, I had served on a workshop with Lee Hood that recommended to NHLBI that they create a program, which they sort of did. It wasn't exactly what we had proposed, but it was designed to have academic centers do exactly what you said, simultaneously partner with industry for projects that clearly had a commercial opportunity, but also to have NHLBI funding those projects that didn't have a commercial opportunity. And the idea was to strike this balance. These were wound up being the National Centers for Advanced Innovation or something. I think that was, and I chaired the steering committee for a while. There was one at Case, I see Pam, one in Boston, and one for the entire UC system. NHLBI ground it down, so it's no longer going on. But I think it was an interesting attempt to try to create a model like the one that you mentioned.
Yes, I think we need to have a long-term program for all 26 virus families that are known to infect humans, and I suppose they could be prioritized, but I don't think we should. I think we should be systematic and go through all of those families and prepare reagents and maybe prepare some prototype products that could be used and have monoclonal antibodies and structures and drugs and the whole thing. So I'm hoping that the event that we just experienced will motivate people, since we did lose $16 trillion. A few $billion a year doesn't sound like as much as it used to sound. And so maybe it'll be possible to have real preparedness as we go forward.
I think Emma had a comment she wanted to make, and Barney, since you've been on both sides, I think it's suitable that she ask you this.
So my comment related, thank you, Barney. Emma Moore from Penn, wonderful presentation, really enjoyed it, and what a monumental amount of work squeezed into an accelerated time point towards the end, really remarkable. My comment relates to cultivating the culture of academic, industry, government, tripartite relationships. And I think part of the challenges within the academy is our reward system is so squarely motivated towards individual accomplishments as opposed to team-based science accomplishments. And when we think about criteria for promotion and tenure tracks in our respective institutions, the jewel in the crown continues to be the R01 grant funding. And certainly the contribution of our clinical investigators is absolutely critical to delivering novel therapeutics into clinical practice. And the majority of these individuals are funded from industry funding sources. So my comment was that part of this narrative comes from the academy itself, and we need to look within to change that particular culture. Thanks again for a great presentation.
Yeah, we were very lucky in this case. And, you know, it felt like all this was being prepared sort of for such a time as this, when this event happened during this last year, because a year and a half ago, nobody really cared that much about coronaviruses. And we just found ourselves at the right place at the right time with the right information and having a way of going forward. But part of that was because of what you just mentioned, and that is we had a monthly call with 4 academic collaborators and with Moderna. And so UNC, Vanderbilt, UT Austin, and Scripps, those collaborators on coronavirus, our coronavirus project that not that many people cared about, we were meeting every month and had very complementary skill sets, and also meeting with Moderna once a month. So we had this government academic industry collaboration that was already preset, and we already trusted each other. And the great advantage, like I said earlier, of the intramural program is that we can make reagents, we can do things that you can't really get paid for on a R01. And so our role was to support the academic sites and help do things that they can't easily get paid for. But getting paid 2 years after you send in your first grant is not conducive to this rapid, fast innovation. And they're really, I hope we can find a different way to fund people who are trying to be innovative and move fast.
Before I turn to, I think Harry Greenberg had his hand up, and then to Al, I just have to ask, you know, I think what we're all hearing you talk about that process, which also had, some help from the warp speed money, but that collaboration, which we're all striving for and we're not getting to, it'd be great if you wrote a process article about that allowed us to understand how that preparedness and the fact that you had those collaborations already formed can help win such battles as this. Maybe that will help. I mean, the R01 is obviously the jewel, mainly, I shouldn't say mainly, it's of course a wonderful thing. It also brings in full overhead to institutions, which they appreciate very much. And I think that if we start highlighting a story like this by its process, not just the outcome, this is a great opportunity to describe that whole relationship, how it all grew way before, because people would be dumbfounded and amazed. I think next question was Harry Greenberg.
There's kind of been this artificial firewall between intramural and extramural NIH, and it is really not very productive. And it's one of the things that I think maybe you're alluding to needs to be torn down a little bit, that extramural, intramural NIH dollars need to mix more than they do now.
Yeah, thank you. Harry? Harry, you are on mute.
Hi, Barney. That was just a fabulous talk, and also some of the best visuals I've ever seen. If you did them, I'm even more impressed. But I wish I had slides like that. I hope they're available, because
I'll send them to you, Harry.
Thank you. They're beautiful. It strikes me now that with the work that you and your colleagues have done, there are new ways of thinking about immunizing people. And one is directly with nucleic acid, RNA maybe, and the other is with very sophisticated protein structures that are really thought out beforehand. And I'm wondering whether, one answer is, well, that one way will suit one pathogen and one way will suit another. And of course, some of this may be used for immunotherapy, not having to do with infectious disease. do you have any prediction about which is the most viable way that people in academia and industry are going to pursue for immunization in the future?
Well, I hope that it's fit for purpose, because I think you're right that not everything is going to be solved by one approach. And in our overall concept for how to deal with these 26 viral families, we've divided them up into entry mechanisms. So the class 1 fusion protein viruses are in one group. The class 2, which are mostly the alpha virus, flavivirus, and bunya viruses, are in another group. And then class 3, like herpes viruses and others, are in another group. And then the non-envelope viruses are in the fourth cluster. And those ways of thinking about grouping viruses, I think, could help work across virus families. And so if you had subject matter experts within those working groups, you could probably accelerate the finding of solutions for some of these other things, because the non-envelope viruses probably need a VLP, whether it's delivered as a protein or an mRNA. I don't think it matters, but some things could use replication defective vectors, like a new adenovirus or a new rhabdovirus if it broke out, and other things need these nanoparticle displays, and some things probably just need a soluble protein. So I think the smorgasbord of choices right now is so amazing. I wish I was 30 years younger, because I think we can find solutions for most of the problems that are out there just from the choices we already have.
Al, I think you had a question next. And then Dan, I saw too.
Okay. Barney, hi. This is Al Reese. I'm from University of Maryland. And fantastic presentation, both yours today as well as your roommate's presentation of yesterday. First of all, there couldn't be a more interesting coincidence with that roommate picture. Was it 50 years ago you said, or something like that? It's
Yes. Yeah.
quite a coincidence. But so that's half of my comment. But the other half is just the spectacular development that has occurred. And I want to ask you a bit of forecasting, because clearly sometimes I say to my faculty all the time, success is for the prepared. And it's clear to me that you and the team were prepared in some regard with regard to the RNA technology that you could actually put in place to achieve, in part, the rapid vaccine development. So my question is, and in large measure, because a good portion of what we do here at Maryland is vaccine development and testing all over the world, do you see that vaccine development will be fundamentally changing to a more nucleic acid RNA-based technology, or it will still just be one additional strategy that will be used? Second question, while I have the floor, and that is, given the fact that we are clearly having emerging infections, it was Ebola a few years ago, and now it's Corona, and so on. Do you think that we were just lucky to have had the RNA technology for the Corona available, and if it were something else, some other virus, we wouldn't be as prepared as we were with the Corona?
Right. So first of all, I want to let you know that Kathy Newsel, there's another coincidence with Kathy Newsel, who's the head of the Center for Vaccine Development at University of Maryland. She was my first fellow when I was at Vanderbilt, our first infectious disease fellow. So I'm very proud of Kathy.
Amazing. Well, I'm proud to have recruited her too.
Yeah, that was a great job. Let me do the last question first. We were particularly well prepared for coronavirus, and coronavirus was particularly well suited for a generalizable approach, because that same set of mutations worked for almost every coronavirus we had tried over those last 6 years. And so we knew exactly what to do, we thought we did anyway, without any additional experimentation. We knew enough to start a program without any additional experimentation. If it had been the Paramyxoviruses, even though we know how to stabilize them, it's a little bit different for every virus. If it had been a Bunya virus, so it would have taken 2 or 3 additional months to get started. If it had been a Bunya virus, we could have been lost for a year or 2 before we even really had the right thing to deliver. And so it does require a lot of preparation. And I think we were particularly lucky with coronavirus, just because, you know, we were, for whatever reason, we had it done ahead of time. And the first question, can you remind me again what your first question
is? Really forecasting, kind of your crystal ball, given the whether or not you do think that we're at a
right.
road.
So I think that this event, like I showed you, there's like every vaccine modality you can imagine has been attempted or used for this coronavirus event. On the WHO landscape slide, you should look at that. It's so I think we've definitely pulled gene-based vectors and mRNA and structure-based protein designs up into, you know, maybe more common use. And so I think that all of those modalities are going to have their purpose. You know, for instance, maybe the gene-based approach for delivery is really good for priming an immune response, but a protein or particle may be better for boosting an immune response. So for something where there was pre-existing immunity, like a new flu, a new influenza comes up, it may be better to use a protein in that case. And there may be other cases where it may be better to use a vector. And some cases, it may be better to figure out a way to deliver it mucosally. And in this case, we're doing well enough, I think, with parenteral administration and protecting the lower airway. But some cases, and Harry, I'm sure, would like to comment on this, if it was a gastroenteritis type of problem or a different type of respiratory problem, you may need mucosal delivery. So I think we need to make it fit for purpose. I think all these technologies are going to be useful in some different types of cases.
Thanks, Barney.
Great.
Appreciate it very much.
I think I'm not very good at this, but I'm monitoring the chat as well. And I think the next question should come from Dan Ford and then Barry Kohler. Go ahead, Dan, with your mute off.
Hi, Bernie. Dan Ford from Johns Hopkins. And again, just celebrating a wonderful achievement for your team. And I think we're looking at the success now. And what I was thinking, if you look back in the last year, at what point did it really take your courage to keep going, where you were like, I'm not sure this is going to work? Or if you think about it, was there a time where, you know, other barriers that you saw that you think for the next time we should be prepared to move even faster?
Well, I wish we had already done the phase 1 trial of an mRNA vaccine with one of those constructs ahead of time, because then we would have had longer term safety data. It would have been the whole process of moving from animal to human trials, and the transition from phase 1 to phase 3 could have been shortened. To me, having that ahead of time would have been the biggest step to have made if we could have gotten to that place. But, you know, I can't imagine what would have happened if this would have been the SARS in 2002. I mean, we didn't have any structures. We didn't know anything about how to design the right thing. We didn't have an mRNA to deliver. It would have been DNA and vectors, and it probably wouldn't have worked that well. And we didn't have Zoom, so we couldn't communicate. So it would have been a totally different story if this had happened in, 20 years ago. And it would also be different if the MERS coronavirus did this, because it has a 35 to 40% mortality rate. So, thank God that it wasn't MERS that did this worldwide circulation, because with a 1 or 2% mortality rate, it almost feels overwhelming. And I can't imagine what it would be with a much higher mortality. So that's the biggest thing. I want to mention something about the way our lab worked too, because I think this is true in many basic research labs, but we have a very diverse group. We have people from almost every continent on our group and in our team, and it's gender balance. It's actually more women than men in our lab, and I think and we also were working with a number of different academic sites. It wasn't just one, it was truly a big team. And so that idea of cultivating a diverse team to do a lot of diverse work and thinking in diverse ways, I think was very important for just getting through, especially those early days. And my only real question was about 2 weeks in, whether I'd chosen the right sequence or not for sure, because now we were doing experiments on other sequences and constructs, and I was saying, uh-oh, I hope I didn't make the wrong choice. But it turned out it was as good or better than anything else. But there was a few doubts ahead of time, but I think the diversity of the team is really part of what made this all work.
Great, that's really helpful.
So
Yeah.
my question goes back to a National Academy committee I sat on after 9/11 that was tasked with coming up with countermeasures to biologic warfare agents. And I learned at that time that there were 18 threat agents, meaning someone had somewhere in the world tried to weaponize those 18 different pathogens. And the question before our committee was, what's the right way to protect against this, especially since there's no naturally occurring infections out there. And there was a lot of, at that time, actually, vaccine development was at its nadir in the United States. And there was even concern that it was going to disappear completely from the United States. I know that sounds amazing, but I can tell you, having lived through it, it was absolutely a real concern. So I was just wondering, and frankly, I came away from that thinking that monoclonal antibodies might be a better choice than vaccines, given the complexity and a variety of other things. But I just wonder, have you guys been brought into that particular area for consideration?
Well, there are ways to make these things even more dangerous. You can put IL-4 or 13 into a pox virus and make it a lot more lethal. So there are ways of doing this, but honestly, I feel like our greatest threats are natural threats, and that the best we can do is to cover the 26 virus families we know can do this. And I'm not a bacteriologist, so there's a lot of other pathogens, obviously, that could be a problem, but I think more about viruses becoming pandemic and rapidly contagious. So I feel like if we really got our head around the options for how to deal with these 26 virus families, then we would be able to adapt. And maybe we need to put an IL-12 into our vaccine to counteract a pox virus that had IL-4 expression or something like that. But I think we need to deal with Mother Nature and not as heavily focused on biodefense. And one of the things that happened after 9/11 was that Dr. Fauci, thank God, was able to turn some of the thinking around the need for more biodefense into programs for emerging infections. So he broadened the scope to not just be biodefense. So all the new funding in NIAID that came during that time didn't just go to biodefense, it went to both biodefense and emerging infections.
Thank you.
But yeah, I would prefer focusing on the natural threats, and because I think that's where most of the surprises come.
You know, this has been so inspiring. Thank you so much for revealing this to us. And also, I think, you know, as a citizen and thinking about the dialogue that's going on in our country right now, I think we understand what a fantastic platform that was built, and the capabilities and the alliances and surmounting cultural barriers, all the things that were brought together to have this work. But I don't think the public understands this in a whit. And, you know, I know Research America does these surveys and finds out that, you know, Americans don't even know what clinical research is, unfortunately, in many cases. I really hope you'll think of making a popular version of this story. I really think this is the thing, like Lewis would write, you know, he can do the big, you know, the big short. He ought to be able to talk about this, because this is really a story of success of in so many ways, science serving society ultimately, but only because of a lot of collaboration. It's just a magnificent story for, as I said in the beginning, for our generational disaster, which only could be answered by clinical research. And by that, I mean translational research from bench through all the way to public benefit. It's remarkable. I hope you'll find a children's book writer or just someone like Lewis or someone to write that.
Well, I have to say in response to that one of our secret weapons is Dr. Corbett, Dr. Kizzmekia Corbett, who is a research fellow in my lab and who's been working with a few students over the last few years on the coronavirus projects. And she has really helped manage the process during this last year of making sure Moderna could keep up with all the things that needed to be done, because most of the preclinical work was done at the VRC. Moderna just didn't have the infrastructure to do that. But, and we got Dr. Corbett out front early on in this process, and she's an African-American young woman scientist, and her voice right now is very important, because there is a lot of the world who can hear her voice a lot better than they can hear my voice, and it's not necessarily just Black people. It goes across races, because, you know, there's something about the youthful way of communicating that is just more compelling. And so I think the other thing that I've learned this year that is still a little disappointing is that we can overcome a lot of these technical things now with the new technologies, but the communication to the public and the preparation of the public for the deployment and the acceptance and understanding of the public is really much worse than I even thought it was. And I wasn't that optimistic to begin with, but it's at a much lower level of understanding than I could have dreamed. And so I think there's a big need for just helping people understand biology better from a lot of different types of voices.
I think until you do the bestseller in the movie adaptation, you haven't finished your job for the public. But on the other hand, unless there's other questions, we're almost at the end of our time. I want to thank you for relating this in such a way that we can understand and appreciate it, and also for the huge amount of work involved. And for all of you here at the business meeting, thank you for coming both here for the business meeting, but also for the centerpiece. Thank you very much, everybody.
Thank you, Harry. Thank you.
Thank you so much for joining us, Barney.
Yeah, thank you all.
Thank you, Dr. Graham.
Bye-bye.
Patty? Let's switch to the Council of Teaching Hospitals. And this, I got my conversation with Benu Kaushal done.
We'll switch you to cough.
Silver, huh?
Now Zoom.